How do specific genes drive or suppress cancer cell growth? Imagine your genes as tiny switches inside your cells that control when they grow, divide, or die. When these switches work properly, your cells grow in a controlled way. But sometimes, these switches get flipped in the wrong direction, leading to uncontrolled cell growth that can cause cancer. This process is especially important in understanding reproductive cancers like testicular and cervical cancer. There are two main types of genes involved in this process: oncogenes and tumor suppressor genes. Oncogenes are like the gas pedal in a car. They promote cell growth and division. Normally, proto-oncogenes are the healthy versions of these genes, helping cells grow when needed. But if they mutate or become abnormally active, they turn into oncogenes. This is like pressing the gas pedal too hard, causing cells to grow uncontrollably. For example, the MUC gene family and parts of the CDK4 and CDK6 systems are common oncogenes that push cells to divide faster. These genes can also help cancer cells avoid dying naturally through a process called apoptosis, which is the cell's way of self-destructing when damaged. On the other side, tumor suppressor genes act like the brakes on a car. They keep cell growth in check by halting the cell cycle when necessary, allowing for DNA repair or triggering cell death if the damage is too severe. When these genes are mutated or turned off, the brakes fail. One of the most well-known tumor suppressor genes is TUP53, often called the guardian of the genome. It helps repair DNA and can initiate cell death if the damage is beyond repair. Mutations in TUP53 are common in many cancers, including those affecting reproductive organs. Other tumor suppressors, like SMAD4, involved in signaling pathways that control growth and death, can also be inactivated, removing important growth controls. Genetic changes that cause these problems include point mutations, where a single DNA building block is changed, gene amplifications, where copies of a gene are increased, or chromosomal rearrangements that shuffle parts of chromosomes. These alterations can turn proto-oncogenes into oncogenes or disable tumor suppressor genes. For example, extra copies of an oncogene can lead to overproduction of growth signals, while mutations that disable tumor suppressors remove the cell's ability to stop abnormal growth. In reproductive cancers like cervical cancer, the role of genes is often linked to infections like human papillomavirus, which can interfere with tumor suppressor proteins such as p53 and retinoblastoma protein. This disruption allows cells to grow unchecked, leading to cancer. Testicular cancer also involves genetic mutations that affect cell growth and death, although the specific genes involved can vary. The overall balance between gene activation and inactivation determines how quickly a cancer grows and how aggressive it becomes. It's also important to remember that the tumor environment, including nearby cells and immune signals, can influence how cancer develops. For example, cancer-associated fibroblasts can promote tumor growth by modifying the surrounding tissue and supporting cancer cell survival. In summary, specific genes drive cancer growth when proto-oncogenes mutate into oncogenes that push cells to divide too much, while tumor suppressor genes normally keep growth in check but lose this ability when they are mutated or turned off. This genetic imbalance is at the heart of how cancers like testicular and cervical cancer develop and progress. Understanding these genetic mechanisms helps researchers develop targeted therapies and improve treatment options. Always consult healthcare professionals for personalized advice and treatment plans, as each person's situation is unique.